Okay. Good evening, everyone. Today I'm discussing about detailed procedure of Elisa and its application. Next slide. Elisa is a basic assay technique known as an enzyme-linked immunoabsorbent assay, which is also known as enzyme immunoassay, that is carried out to detect and measure the antibodies, hormones, peptides, and proteins in the blood. Next slide, sir. Types of assay. There are 4 types of assay: direct ELISA, sandwich ELISA, indirect ELISA, and competitive ELISA. Direct ELISA are the antigen coated to multiple plate is detected by the antigen antibody that has been directly conjugated to an enzyme, that we call it as an direct ELISA. The indirect ELISA is an antigen is coated to the microtator well. So sandwich ELISA antibody is coated with the microtator well, and competitive ELISA says microtator well which is antigen coated is filled with the antigen antibody mixture. Next slide. So this is a structure of ELISA which includes proteins, antibodies, and antigens. Next slide. So this is a procedure of ELISA. So here the antibodies and antigen and the proteins includes us in the procedure of ELISA. So the antibodies attached to the polyester plate, which is solid surface and is attached or as a affinity towards bacteria and antibodies and hormones. The microtator coated with an antigen is filled with this antigen antibody mixture, after which free antibodies are removed by the washing. Sir, go to the picture only, sir. I'm explaining the picture. Yeah. The second antibody specific to primary antibody is added with which is usually conjugated with an enzyme. The free enzyme linked secondary antibodies are removed by washing the plate. Finally, the substrate is added. The substrate is converted by the enzyme to form a colored product, which can be measured by the spectrophotometer. Next slide, sir. Next slide. Next slide, sir. Procedure explanation. The disease diagnosed using ELISA are Ebola, AIDS, rotavirus, Lyme diseases, toxoplasmosis, Zika virus, and carcinoma and epithelial cell. Next slide, sir. The applications of ELISA. The ELISA can be used in the food industry. ELISA can be used in the food industry, vaccine development, immunity diagnosis, and pregnant test. In food industry, the ELISA plays a major role in the platform identification of food allergens in such as milk, peanuts, walnuts, almonds, and eggs. It also plays a very important role in the applications of important role in the identification of the vaccines and diagnosis. And ELISA plays a major role in the diagnosis of dengue fever, HIV, influenza fever, and other diseases. And even it plays a very important role in the pregnancy test also. Thank you, sir.
Abhilasha?
Thank you, sir.